Okay, can somebody confirm you can hear me?
I can hear you.
Thank you, Haley. Let me share my screen. Let me state that I did do some grading last night. So besides the grading I got done this weekend and Monday, I've now graded lab 00 and lab 01. I think I got the both of the quizzes, not the quizzes, let me stop sharing my screen then explain this a little better. I'll call up the grade book and then I'll remember what the things I did. The quizzes, the practice quizzes got graded. I did those actually earlier than last weekend. I'm not sure when I did those, but those have been graded. Practice quiz 00A, you'll have to click on it to see it. It is graded, but on most of you, it's not showing. So you got to click on it and then it'll show. There's a problem with practice quiz 00A, and for some reason it doesn't show the grade in it. It's like it's a new submission, even though I got it graded. It must be a bug in the program or something. I've seen this before. I might have done extra credit study 2 last weekend. I don't remember. I definitely did worksheet for lecture 3 last weekend. And then I got the last lab graded, lab 4, the first time. And then for the second time, I did lab worksheet for lab 00 and lab 01. So those two labs, 0 and lab 00 and lab 01, have now been graded for both times. Any question about any of that? Let me get out of the grade book here and then I'll reshare my screen. All right.
I have a question. For the lab 00, do you leave like a little comment about what you miss or what I miss, or because I know the first time I missed one question, I believe, and then it was graded again, and then you didn't leave a comment, so I just wonder if
I usually leave a comment the second time I grade saying what was wrong. If I didn't do that, I apologize. Can you send me an email and I'll go in there and correct that?
yes, thank you so much.
All right, thank you. All right, am I sharing my screen? No. All right, now I am. So today we're going to talk about lab 6. Today is May 2nd, and we're going to talk about lab 6, disc diffusion antibiotics. Let me tell you, you also need to read about the unknown project instructions. You can find that file on Canvas. You need to have that read by next Tuesday, because I'm going to start the unknown project on Tuesday, and I will talk about it, but it would be helpful if you would at least look over the unknown instructions. And let me see if I can show you where that is. Scroll down. I think it's week 5 lab. There is week 5 lab. Unknown report. I have a sample journal, what your lab report will look like. And there's the unknown instructions. You should read this file by next Tuesday. The other files you don't need to look at yet, but I will talk about them next Tuesday. And let's see. Yeah, you don't need to read any of the others. You can maybe read about the grading report if you want, but you should do this before you turn in the report. Look at the unknown report grading, building a dichotomous game plan, and obviously the unknown instructions and the sample journal. As you're working on the unknown report, you should look at your unknown notes pages and the unknown list. You can't work on your report without working on these two pages. Any questions about any of that? I will talk about your unknown report next week, next Tuesday. All right, let's talk about Lab 6, disc diffusion and antibiotics. Lab 6, disc diffusion, evaluation of disinfectants and antiseptics to antibiotic and antibiotic effectiveness. There is some reading in Tutora, page 177 on terminology, page 187 to 194 on principles of effective disinfection in evaluating a disinfectant, page 196 to 197, so one page on chemical methods of microbial control, another page 554 to 555 on antibacterial drugs that we normally call antibiotics, and then 2 pages 567 to 569 on tests to guide chemotherapy, the disc diffusion method. There is some supplemental material in today's lab. There's video clips and a virtual lab. Oops, too far. The learning objectives to this lab: first, be able to identify the type of chemical agent and the active ingredient used in various disinfectants and/or antiseptics. 2, evaluate the effectiveness of disinfectants or antiseptics using the disc diffusion method. 3, determine the mode of action of various antibiotics. 4, evaluate the effectiveness of various antibiotics using the disc diffusion method. 5, and actually on 4, this will be the main part of this lab, so you should know 4 very well. 5, list the variables that influence antibiotic disc diffusion susceptibility testing. 6, be able to define the terms. What's sterilization, disinfection, a disinfectant, an antiseptic, a bactericidal agent, a bacteriostatic agent, what's an antibiotic, what's a spread plate, and what is a zone of inhibition. Any questions about any of the learning objectives? All right, evaluation of disinfectants and antiseptics. Sterilization refers to the elimination of all viable organisms, including endospores, that may be present. Something is either sterile or it's not sterile. Disinfection refers to the destruction of vegetative cells, not necessarily endospores. Disinfectants are chemical agents applied to an inanimate object. So when you use bleach on the toilet, we call the bleach a disinfectant, disinfecting the toilet. However, if I were to use bleach on a cat, then it would not be called the disinfectant, it would be called an antiseptic. An antiseptic is a chemical agent applied to living tissues. Usually, an antiseptic is less harsh and maybe not as powerful as a disinfectant. And that's why we don't usually use bleach as a disinfectant, because it's irritating to the skin. But at least an older treatment of necrotizing fasciitis was to wrap the patient their skin, the area infected, with gauze with bleach. So it used to be, at least an old treatment for necrotizing fasciitis. And of course, you also treat the patient with antibiotics. Any question about any of that? All right, to evaluate the effectiveness of a chemical agent, the surface of a nutrient agar plate is inoculated with the particular bacterium that you're testing. Paper discs are then saturated with different chemical agents that are applied to the surface of the agar. The chemical agent will diffuse into the agar, establishing a concentration gradient around the disc. You then incubate the plate overnight and pull the plate out and look at the bacterial growth pattern around the disc. If a zone of inhibition forms, a zone of inhibition is an area of no bacterial growth, the chemical can be assumed to be at least a little effective for that particular bacterium. Conversely, if no zone of inhibition appears, the chemical is assumed to be ineffective. Any question about any of that? All right, table 6.1 tells you the different chemical agent, their general action, and their preferred use. Like ethanol is an alcohol. It can denature proteins and dissolve lipids, which will kill cells. And it's used as an antiseptic. However, it's a very weak antiseptic, so we usually call it a degerming agent, something that has antiseptic properties, but it doesn't remove all of the bacteria from the region you treat with alcohol. So we call it a determining agent. Aldehydes, like formaldehyde, you don't need to know the general action, protein denaturation, usually used as a disinfectant. This is a good disinfectant. It's one of the few that kills endospores. Chlorine bleach, and iodine or halogens. They're used, iodine as an antiseptic, bleach usually as a disinfectant. Peroxygens, like hydrogen peroxide, is an antiseptic, and it doesn't say that right here, but it's actually a very good antiseptic agent, and it does kill endospores. Any question about any of that? All right, let's move on. The zone of inhibition size depends only partially upon the sensitivity of the organism to the disinfectant or the antiseptic. There are various other factors that affect the zone size, including the concentration of the chemical. Generally, the more concentrated the chemical, the larger the zone size. The molecular weight of the agent. Generally, the larger the molecular weight of the agent, the less the agent will diffuse, and the smaller the zone of inhibition. And then the density of the bacterial inoculum can also affect the size of the zone of inhibition. Depending on what your concentration is, if you increase the concentration of the bacteria, then generally the zone of inhibition size will be smaller. Therefore, we have to standardize all of these variables in order to get a reliable measure of the sensitivity of the organism to the antiseptic or the disinfective agent. Evaluation of antibiotic effectiveness. Antibiotics are categorized as bactericidal if they kill the bacteria. They are characterized as bacteriostatic if they inhibit the growth of the bacteria. When a bacteriostatic antibiotic is used, we are relying on the immune system to assist in the elimination of the infection. because a bacteriostatic agent will not kill the bacteria. It'll stop it from growing, but once you stop applying the bacteriostatic agent, the bacteria can then regrow. Any question about any of that? All right. The Kirby-Bayer diffusion method, also called the diffusion method, is the most common method for testing for antibiotic susceptibility. The results of such testing help physicians in choosing which antibiotics to treat a sick patient. Bacteria are isolated from the patient, grown on an agar plate, or put on an agar plate, and then the paper discs impregnated with an antibiotic are applied to the surface of the agar. That will establish a concentration gradient of antibiotic around the disc. You then incubate the plates, pull out the plates, and then look for a zone of inhibition. Where the antibiotic concentration is high enough, you'll have inhibition of the bacterial growth, or a zone of inhibition. Now remember, this may not kill the organism. It may only inhibit the bacterial's growth. So here we're looking at the Kirby-Bayer susceptibility test. Let me blow this up a little bit. And you can see that some of them have a very large zone of inhibition. That's fairly large too. Some of them have a smaller zone of inhibition. That's real tiny zone of inhibition. And that one is probably no zone of inhibition. This is kind of curious. Hopefully you'll never see that. That looks like there was originally a zone of inhibition, and then the bacteria started growing. So that there's a ring, but then bacteria are inside that zone of inhibition. Like I said, hopefully you'll never see that. It might be just the plate is foggy, because that one kind of looks white too. In which case that is a zone of inhibition. Like I said, hopefully you'll never see that. That might be just a picture, an artifact of the picture. All right, so which would be the best antibiotic to use on this patient? Anyone want to guess? I will warn you, it's a trick question. Okay, I was expecting an answer. You guys are not very helpful. Some students would say this one has the largest zone of inhibition, so it would be a good antibiotic to treat with the patient. However, you have to take into account some other factors, like the molecular weight of the antibiotic. If this is a very small antibiotic, and this is a very large antibiotic, we expect a larger antibiotic to diffuse less in the agar than a small antibiotic. And that's why this zone of inhibition may be larger than that one. So when we're talking about what is the best antibiotic to look, you can't just measure the size of the zone of inhibition and say this is the best one to use. You have to take into account other factors. Any question about that? And the main one is the size of the molecular weight of the antibiotic, but there are some other factors as well. Any question about that? It's an important part. The inhibition zone size is dependent on several factors: the diffusion rate of the antibiotic, the degree of sensitivity of the microorganism, and the growth rate of the bacterium. Table 6.2 lists the procedural protocol required to standardize these variables when performing the Kirby-Bayer disk diffusion tests. So the standardized protocol for Kirby-Bayer disk diffusion procedure is 1, you inoculate nutrient broth using several bacterial colonies isolated on a non-selective media. And the bacteria should be no older than 24 hours old. The reason is you want quickly growing bacteria, and how to get that is to have bacteria that is fresh. You then grow up the broth to a turbidity of 0.5 McFarland standard units. That's approximately 1 × 10⁵ organisms per mil. And if the turbidity is higher than that, you then reduce, you can dilute it and get it 0.5 McFarland units. If the turbidity is under this, then you have to grow it longer. Any question about any of that? So you always apply broth turbidity of 0.5 McFarland units. You then inoculate a dish at room temperature, and that dish is always Mueller-Hinton agar between pH 7.2 and 7.4. If you use another media, you can get different results. So you always use Mueller-Hinton agar between pH 7.2 and 7.4. And obviously, the pH can affect the size of the zone of inhibition. If you had it too acidic or too basic, you won't get the bacteria to even grow. The agar depth is always 4 millimeters deep. The deeper the agar, the less the antibiotic will diffuse on the plate. So when we do the Mueller-Hinton, not the Mueller-Hinton, the Kirby-Bayer test, also called the disk diffusion test, we always put it on plates that are exactly 4 millimeters deep in agar. The plates are always 150 millimeters plates. And then we apply the bacteria with a non-toxic swab using the spread plate technique to obtain confluent growth. And I'll talk about that when I go through it in the lab later. You then place the disc on the agar 5 minutes after you have placed the bacteria. You allow a little bit of time so that the liquid that you apply the bacteria to the plate gets absorbed by the agar plate. So 5 minutes then later, you place the disc on, placing each disc at least 24 millimeters from each other disc and no more than 12 discs per 150 millimeter Mueller-Hinton plate. Oh, right there it tells you add the antibiotic disc 5 minutes after putting the bacteria and no longer than 15 minutes after adding the bacteria. You then let the plates sit for no longer than 15 minutes after adding the antibiotic disc before incubating them in a non-CO2 incubator for 16 to 18 hours at 35 degrees C. We always grow them for only 16 to 18 hours because the different growth will have a different zone of inhibition, always at 35 degrees C. Any question about the standardized procedure for doing the Kirby-Bayer protocol? When you're doing the Kirby-Bayer protocol using the standard procedure, you should have similar, if not the same, zone of inhibition sizes for each person doing the Kirby-Bayer procedure, wherever they are located on the planet Earth. Any question about any of that? After incubation, the inhibition zone sizes are then measured with their diameter using millimeters. You then look up in the table made by the National Committee for Clinical Laboratory Studies, abbreviated NCCLS, that sets its standards for interpreting the zone sizes, whether the bacteria is considered to be sensitive, intermediate, or resistant to that particular antibiotic. For example, when we're looking at the antibiotic penicillin G applied to the bacteria Staphylococcus aureus, we grow it up and we have the zone of inhibition there. There's the paper disc. We then measure the diameter of the zone of inhibition, and it looks like it's 20 millimeters. We then look at that zone of inhibition size in the NCCLS table of standards, and it'll say that for the antibiotic penicillin G for Staphylococcus aureus, if the zone of inhibition is 20 millimeters or less, then that bacteria, Staphylococcus aureus, is resistant to that antibiotic penicillin. If, on the other hand, the size of the zone of inhibition is between 21 to 28 millimeters in size, then the zone of inhibition is intermediate. To be sensitive, you have to have a size of the zone of inhibition to be greater than or equal to 29 millimeters. So that for this particular strain of Staphylococcus aureus, to penicillin G, this strain is resistant to penicillin G because the size of the zone of inhibition is 20 millimeters. 20 millimeters or less, the NCCLS table says it is resistant. Any question about that? So how you determine whether an antibiotic is sensitive or resistant, you have to do the Kirby-Bayer test and then measure the size of the zone of inhibition and then take a look at that measurement, the diameter, compared to the NCCLS table of standards. And there will be a number for resistance for each species, in this case Staphylococcus aureus, for each antibiotic. And they can be different for each species and then for each antibiotic. Any question about any of that? All right, know the terms of this lab. In this laboratory, you're not going to be physically performing any of these activities, but you will be held responsible for how to interpret the effectiveness of antibiotics using the Kirby-Bayer disc diffusion method. In procedure 1, we have a video clip you should watch on preparing a spread plate, and then you should review the steps for using aseptic technique when preparing a spread plate or a bacterial lawn of growth. Let me go ahead and show you how to do that. We're going to pretend that this is a cotton swab, which maybe I should use this edge better, and that this is a petri dish with agar on it. Obviously, it's sterile agar plate. Mueller-Hinton between pH 7.2 and 7.4, etc. How you get the bacteria to grow everywhere, what we call a lawn of growth, is you get your stock culture, which is a broth at 0.5 McFarland units of bacteria, and you stick the swab in the broth, and then you touch the side of the tube inside the tube. I don't want to do it inside because the pencil isn't sterile and it's in my antacid tablets, but you stick it inside and then you touch the side of the tube inside the tube to get the worst of the liquid out so it's not dripping. And then you take your agar plate, uncover it obviously, and then you swab everywhere on the agar plate. I don't know if you can see this, but I'm trying to swab everywhere. And obviously this isn't a circular plate. Then you rotate the swab a quarter of a turn, and then you rotate the plate a quarter of a turn. And then you swab everywhere again. Then you rotate the swab a quarter of a turn, and you rotate the plate a quarter of a turn again. And then you swab everywhere one more time. You then take the swab and then you move the swab along the edge of the plate. I don't know if you can see that. And that's because when you're swabbing, sometimes you won't get the entire edge. You want the bacteria to grow everywhere. So you do this, the swab on the edge. It's important that you get the bacteria everywhere so it grows up everywhere, and we call that a lawn of bacteria. I once had a student who went something like, oh, oh, let's just go like that. And then she came and showed me the plate, and it had very large holes of areas, which she never swabbed. So there's no bacteria in very large parts of the plate. And I looked at that and I said, you didn't swab everywhere. You were supposed to swab everywhere. Because of the large holes, we can't measure the diameter of the zone of inhibition around the disc. So I told her she could either redo it or she could use her steady buddies or her partners' plates because she couldn't do the lab on her plate. Any question about that? You then allow the liquid to absorb for 5 to 15 minutes before you put on the antibiotic disc. And then you put the plate in the incubator upside up and then incubate it. When the time is done, I think it's 16 to 18 hours, you then pull it out of the incubator and look at the plate. So procedure 2, we're going to evaluate the effectiveness of antibiotic treatment of the Kirby-Bayer test. View the following video clips and complete the virtual lab. So we have a video clip on preparing a culture for the Kirby-Bayer disc diffusion. And then we have a video on interpretation of the results of the Kirby-Bayer disc diffusion. And then we have a virtual lab on the Kirby-Bayer disc diffusion method. Let me go ahead and open that. This may take a little bit of time to load depending on how fast your internet connection is. You can see I've got a fairly fast connection. You click start, you can click the other things, but you can click start. And here we have the Kirby-Bayer antimicrobial susceptibility test, the disc diffusion method, with E. coli. And there's the date. What you do is you take the ruler here and bring it up to the diameter. You have to move this exactly where the diameter is and then make a measurement. So that looks like maybe 17, 18 millimeters. That's TE. TE is tetracycline. And then I said 17 to 18. So if it's green, you guess correctly. If you get incorrect, you can see a red X through it. Red X through it. So like streptomycin, that's incorrect. That's incorrect. So it's 15. But go ahead and do this. Obviously, with CC and E, you have no zone of inhibition. So that would be less than 13 for E. All right, any question on how to do this? Now hit the menu again, hit restart, and it'll show you the plate for Pseudomonas aeruginosa. Once again, take the ruler. Don't need to do it for CC because once again, it's 0. What is CC? Clindamycin. But you do it for C, which is chloramphenicol, and that looks like 16. So that should be intermediate. Any question about any of that? And then hit the menu again, restart, and we get our third plate, Staphylococcus aureus, with different antibiotics, different zones of inhibition. In this case, this looks like this might be MRSA, multi-drug resistant Staphylococcus aureus, because there's only one antibiotic which is showing a zone of inhibition, and that's vancomycin, which, if you don't know, is how they usually try to treat MRSA. As long as it's sensitive to vancomycin, they'll give you that. And that looks like it's about 17, which is sensitive. Any question about any of this? All right, so remember to get the different plates, you got to click on the menu button and then hit restart. It goes back to the first one. Hopefully I didn't close down the wrong thing. Okay. You then, after you get the measurements for the Gerber Bayer susceptibility test, you then take that diameter measurement and look it up in the table of the NCCLS zone standards for interpretation. So here we have, I think this is E. coli, is that right? Yeah, E. coli. For the zone of inhibition to ampicillin, the zone of inhibition to cephalofithin, the zone of inhibition to gentamicin, and the zone of inhibition to tetracycline. You use the calipers to measure it. We don't have a ruler, but you can use the caliper. Note how to read the caliper. You go from 0 to 0. So this is a hair shorter than 15 millimeters. We'll call that 15 for ampicillin. You do not read from here to there because this 0 mark is not over here. You read from 0 to 0. Any questions about any of that? So use the caliper, you read from 0 to 0, and this would be about 15, a little yes, just a hair less than 15 millimeters. And then take a look at the table. For E. coli, for ampicillin, 15 is right there. And then that's the sensitivity of E. coli to ampicillin. Go ahead and do that for each of the antibiotics shown here. And then fill in the table. I've already done the first one for you. Ampicillin, AM, the zone of inhibition is about 15 millimeters. The interpretation, I gave it to you earlier, but put it in there. And then do the same for cephalofithin, gentamicin and tetracycline. Give me the measurements of the zone of inhibition and their interpretation. For full credit, you must be correct on each of these. Otherwise, you'll get partial credit. Everyone should get the first one right because I gave it to you. Although I'm not writing it in here. So fill in the table. I thought this had a number. Maybe that's number 2. And then on number 3, list the variables that must be standardized when performing the antibiotic susceptibility test, meaning the Kirby-Bayer diffusion method, using, well, there it is, the Kirby-Bayer diffusion method. And we just mentioned those in the lab. So you need to fill in 1, 2, 3, 4, 5 different things which are standardized. Question 1. The size of the zone of inhibition often varies for 2 different species of bacteria when challenged with the same antibiotic. What is a possible explanation for these differences? Write your answer here. 2. What would happen if we didn't use aseptic technique to put the antibiotic disc on the plate? What happens to the results of the test? All right. 3. What would happen if we increased or decreased the concentration of the antibiotic in the disc? So obviously that will affect the size of the zone of inhibition. So what will happen? 4. Antibiotics can be bactericidal, that is, they kill the microbe, or they could be bacteriostatic. They inhibit the microbial's growth, but do not kill the bacteria. When observing a zone of inhibition around the disc, how would you determine whether the antibiotic causing that zone was bactericidal or bacteriostatic? Any questions about any of that one? So how can we tell whether the bacteria here is going to be killed by this antibiotic or whether it's just the antibiotic is just stopping the bacteria's growth? Anyone have a guess? All right, think about that one. I'm not going to give you the answer because that's the question in the lab. Oh, this is going too slow. Actually, we should move to the worksheet anyways. Remember to submit the worksheet. Do not submit the lab module. You'll get a warning or a penalty if you submit the lab module. Four. The size of the zone of inhibition often varies for 2 different bacterial species. Oh, I've already talked about that, sorry. 5. What would happen if we didn't use aseptic technique to put the antibiotic disc on the plate? Mention what will happen to our test results. 6. What would happen if we increased or decreased the concentration of the antibiotic in the disc? Be specific in your answer. 7. Antibiotics can be bactericidal, that is, they kill the microbe, or bacteriostatic, they inhibit the microbial growth, but do not kill the bacteria. When observing a zone of inhibition around a disc, how would you determine whether the antibiotic causing that zone was bactericidal or bacteriostatic? Question 7 is a thinking question. Most students get it wrong the first time. So you need to think about it before you attempt to answer that one. Most students actually do get question 7 on the second grading. Not all, but most. When performing the antibiotic sensitivity testing, it's important that the test conditions are kept very standardized, meaning the same type of media, Mueller-Hinton, the same agar death, the same age of the culture or similar age of the culture. Why do we do that? 9. Are there any other substances besides antibiotics that we could evaluate using this Kirby-Bayer disc diffusion method? And then name one. 10. Does the presence of a zone of inhibition necessarily mean that the bacterium being tested is sensitive to the antibiotic? Explain your answer. 11. From the size of the zone of inhibition of chloramphenicol on E. coli in the virtual lab, is the bacteria sensitive, intermediate, or resistant to that antibiotic? Then for chloramphenicol on P. aeruginosa, is the bacteria sensitive, intermediate, or resistant to the antibiotic? Any question on any of these?
Question. Did you say there's 11?
Yes.
Number 11? In the module and in the worksheet, I cannot see that.
There should be a number 11 in the worksheet. You're not seeing that?
No, I printed mine, but it
Okay,
starts number 2.
I probably pulled this from last spring. Thanks for letting me know. I'll download or upload a new worksheet for this lab.
And the module 2 is different.
Yeah, the
like-
module is not going to have it. I'm not going to redo the module, but the worksheet will have question number 11 on it. Okay, thanks for
and it
letting me know
starts
that.
number 2.
I'm sorry?
The worksheet starts number 2. There's no number 1.
Is it the same one?
It says worksheet lab 6, and it starts with number 2. I don't know if
Uh,
I'm only the one. I don't know.
all right.
Somebody got
I'm going to assume it's the same as this worksheet here, and I'm going to upload this worksheet actually. Let me get this out of
I
there.
would love to see what you're reading.
I'm got it right here. Should be able to see it now. All right, let me upload that. Are there any other questions? If not, I'll be here until 8 o'clock to help you with any questions on the lab, or until the last student logs out. All right, let me stop sharing. Go ahead and shut that down. Am I not sharing my screen? I meant to
No,
be sharing my
You
screen.
did not. You did
Ah,
not share.
Yeah, when I'm not sharing my screen in the lab, just let me know. I meant to be sharing my screen.
Yeah,
Okay.
It-
Okay. All right. Let's stop the recording. Something's different about this. It's totally different